Well, aloha everyone, and welcome back to Biology 101 here at Chaminade University. This will be my last lecture for Biology 101 because this is Unit 14 and it's the very last lecture of the semester. However, it is a pretty long one and so it'll probably get split into two. What I would suggest is that you pause here, go get yourself a cup of tea and settle in and listen to this lecture more along the lines of a story in terms of listening to the history of life on Earth. So now that I presume that you've got your cup of tea, we're going to continue on. Welcome to Unit 14. To begin with, we'll talk about the start of life. So how did life arise on Earth? We'll talk about the very earliest organisms, which were prokaryotic organisms, and how they then became multicellular organisms over many, many millennia of selective pressure. Then we'll talk about how life, which was created in the water originally, invaded the land, and then we'll talk about multiple different sets of extinctive events which have provided selective pressure in the history of life. And then eventually, at the very, very end, we'll talk about the midnight emergence of humans and how humans evolved on our planet. So, up until the 19th century, before Darwin, we really believed that all of the members of a species just survived, were created by spontaneous generation, and that it was created actually from unrelated, non-living life forms. Because at this point, we're talking about an era where the church is funding all of the scientific research, and hence the main dogma at the time is that God created all life on Earth. And a lot of these medieval beliefs were reflected the concept of spontaneous generation. So it wasn't just the idea that all life was created by God at one given point, but God actually was still creating life as they knew it. For example, meat that was left out to rot had maggots that spontaneously were generated from it, and broth that was left out for too long spontaneously went rotten with microbes. Additionally, mice were thought to arise from the combination of sweaty shirts and wheat that had been left out, and so if you didn't keep your silos clean, then you would get mice. And it wasn't until the mid-1600s that Francesco Redi discouraged the idea that maggots actually came from meat. He demonstrated that if you covered the meat with gauze and therefore the flies weren't able to get to the meat, then you were able to keep the meat maggot free. But if you recall, that was one of our very earlier lectures when we discussed that experimental design. Then Louis Pasteur came around, and Louis Pasteur made leaps and bounds of scientific progress. But he is one of the first people, in fact, the word pasteurize comes from Louis Pasteur. He's one of the first people to investigate microorganisms. He's also the person who created the rabies vaccination. He actually injected himself with the vaccination because it was the very first vaccine around, and no one had any trust in science at the time. So then he injected himself with the rabies virus and did not contract it. Side note, this was an era when science was wild, wild west. Anyway, Louis Pasteur demonstrated that if you were able to sterilize the broth, microorganisms did not ever appear until the broth was exposed to existing microorganisms in the surrounding environment. And this is the concept behind pasteurization, right? High heat, high pressure, you kill everything off, and then if you prevent anything from getting into, say that milk, for example, and then you can have an increased shelf life because microbes aren't available to be able to get into the product to be able to consume it. And so that was the final nail in the coffin for the notion of spontaneous generation. But that didn't actually address where life came from. And there's a famous quote by Stanley Miller stating that Pasteur never proved it didn't happen once. He only showed that it didn't happen all of the time, which is true, right? Just because it didn't arise in this particular moment in that broth doesn't mean that that's not where it came from originally at one point on the planet. But this is the refutation of spontaneous generation, and you can see that if you sterilize the flask by boiling it, and see this is an S-shaped flask, and so if any microbes do come in, they're going to get caught here, right? So microorganisms could possibly get in, but they wouldn't be able to get up and over, and that means that we would have no growth even though it's exposed to the air. So it's not just something that spontaneously generates. You actually have to break that flask off in order to allow the microorganisms into the broth in order to see the growth. Basically, you have to inoculate this medium with whatever microbe it is that's going to end up growing inside there. And more modern ideas about the origin of life started to arise in the early 1920s. Both scientists in Russia and in England, so Oparin and Haldane, demonstrated that spontaneous formation of the complex organic molecules that are necessary for life today would not be permitted in the atmosphere that we currently live in. Today's atmosphere is really rich in oxygen, and it just simply won't, because oxygen is a kind of a destructive molecule, it simply wouldn't be able to happen. However, keep in mind that the early Earth had a lot less oxygen than we do now. So oxygen, as I mentioned, it's kind of disruptive. It readily reacts with other molecules, disrupts all sorts of chemical bonds, and if we have a lot of oxygen in our environment, we tend to break things down or keep molecules simple. And you actually have seen this multiple times in your life. Anytime you've purchased something that's been vacuum sealed and that, for example, lunch meats has lasted for a very long time in the vacuum pack, but as soon as you open it, if you lift it open for that same period of time, you would see decay, right? And that's because there's oxygen present, and oxygen is what microbes need. And also, oxygen itself is kind of a volatile molecule. It's going to help break down complex molecules, basically. However, early Earth actually had far less oxygen than we have on our planet today. And that low oxygen environment would have made it that we were able to have the spontaneous formation of complex organic molecules that we currently are not able to have because of the fact that oxygen has a high reactivity with chemical bonds. Um, and so, in order for this complex organic molecules to have come into being, it would have had to have been in a low oxygen environment. And this is what we refer to as basically the chemical version of survival at the fittest. So this is a prebiotic life. This is life before life, if that makes sense. It's all of these different small molecules that are kind of bouncing off of one another and interacting with one another and becoming more and more complex molecules. And this is basically the evolution of life from non-life. And we've actually demonstrated this in the mid 1950s. We were able to create life, so the spontaneous generation, from something called primordial soup. Basically, what has to happen, again, we have to eliminate oxygen, so we have low oxygen conditions, and we're going to emulate the atmosphere of early Earth, so lots of methane, ammonia, hydrogen gas, water vapor, and then some sort of spark or electricity. So if you mix all these gases in a flask and then you add electrical discharge to simulate lightning or some sort of electricity, eventually you get simple organic molecules. You get actually simple organic molecules after just a few days, and you get more complex organic molecules in a week or so time. And this is actually something that most high school classrooms are able to emulate today. So we do know that spontaneous generation of life from non-life does occur. And when I say life, I mean DNA and RNA. I do not mean cells, right, or multicellular organisms, but you have to imagine that RNA and DNA were the basis of where life began. And so in order to be able to emulate that, it's really interesting to know that we're able to do this just in a flask. And again, that is called the primordial soup experiment, and it demonstrated that you can have small molecules forming into larger organic molecules in that early atmospheric conditions if we have electrical energy present. So really all you need, methane, hydrogen gas, water vapor, electrical energy, and ammonia. All right, so that's what this looks like. This is the spontaneous generation of life. Basically, you have water, and this water is subjected to heat, so that means the molecules are bouncing off one another inside water, and then they boil and come up through this chamber and then end up going down into a chamber where there's electricity, and they're able to interact with, so we have water vapor interacting with hydrogen, ammonia, methane, and then it condenses down through a condenser, and then every so often you take little samples of that water, and over time you start to notice that we end up getting organic molecules in this in the samples that you take from that water over just a couple of days. And that demonstrates that organic molecules can form under spontaneous conditions, basically, into what we call prebiotic conditions. That produces things like amino acids, proteins, nucleotides, ATP, and other molecules that we consider to be characteristic of life. Now, again, we have definitely demonstrated that early atmosphere is different from the atmosphere that we see today. However, we've gotten more precise over time, and modern geochemists now believe that the early atmosphere is a little bit different from what they modeled in their experiments. And so we've done more experiments, so additional experiments with a little bit more realistic, still in the absence of oxygen, where the atmospheres are a little bit more like what they truly believe to have occurred in that time frame, and that has also yielded organic molecules. Additionally, we've demonstrated that it's not only electricity that's a suitable energy source, UV light and heat have also been demonstrated to drive the formation of organic molecules in similar experimental conditions. Additionally, we now know that organic molecules are very likely to have arrived from meteorites and comets, so from outer space, literally. We've demonstrated that present day meteorites that have been recovered from regions on Earth that have had a greater impact have demonstrated high concentrations of amino acids and other simple organic molecules. And we've also demonstrated that in space-like conditions, very low temperature, very low pressure, and bombardment of UV light, we can take small molecules and create larger organic molecules from them. So again, we're demonstrating that life can arise from non-life as long as they have the correct starter blocks, low oxygen, and the correct electrical conditions, or UV light or some sort of source of energy. Um, However, we are speculating that prebiotic synthesis is probably very inefficient and also relatively slow, and that prebiotic molecules were probably threatened by a lot of things, including UV radiation. Please remember that early Earth had no ozone layer, right? We had no oxygen. Oxygen is what creates that ozone layer, or very little oxygen. And so the UV energy coming from the sun was probably very raw. Again, that ozone layer that surrounds the atmosphere today is enriched with ozone, and that comes from um, oxygen molecules that are binding with, so O2 molecules that are binding with individual oxygen molecules. So ozone is actually O3. Okay, so oxygen atoms react with O2 molecules to form O3, which is ozone. Now, before we had the ozone layer, UV bombardment from the sun must have been brutal. And we know that UV radiation could provide the energy needed to create these organic molecules, but it can also have the energy to destroy them, or very destructive energy to be able to pull them apart. And that means that very likely the prebiotic conditions that would have been right to be able to create organic molecules would probably have been under protective conditions, areas underneath rock ledges or at the bottom of a shallow sea. So a little bit of protection from UV radiation, and that would allow the accumulation of organic molecules in those regions. Perhaps clay or some sort of silt would have held these molecules down. So one of the lines of thought at this time is that small molecules accumulated on clay particles that could have possibly had small electrical energies. So that would have been like a magnet that would have pulled molecules of the opposite charge down and held them in place, into position, and allowed them to interact with other molecules that may have washed across them. And originally, while these molecules formed on clay at the bottom of the early Earth, etc., bottom of the ocean of early Earth, I apologize, they've now become the building blocks of the living organisms, the first life on Earth. And it's very likely that the first self-reproducing molecule was RNA and not DNA. RNA is a little bit more simple, and it's not as complex in terms of replication. So DNA replication requires large, complex proteins and enzymes that are specific for DNA replication. And also, instructions for building these enzymes are coded in DNA. So in order to create the DNA, you would have had to already have the DNA, which seems pretty complex. So it's more of a chicken requiring an egg situation. So that makes DNA a very unlikely candidate for self-replication. However, it's very possible that our current DNA-based system evolved from an earlier system that was RNA-based. We now know that RNA can act as a catalyst for RNA replication, so RNA is a good candidate as a self-replicating informational molecule. And in the late 1980s, scientists Czech and Altman were working with an organism called Tetrahymena. And they discovered, using this organism, a reaction in the cell that was catalyzed by an RNA molecule. And that means that RNA, it's called a ribosome, the RNA molecule can actually catalyze other RNA molecules. And that means that RNA is able to act as a catalyst to cut other RNA molecules in pieces and able to splice together different RNA fragments. And we now know that ribosomes also are able to, in fact, we know that that's the site of amino acids connection, right, of protein synthesis. And so that means that RNA can play multiple different roles, which makes RNA a very likely candidate as the first self-replicating molecule. In fact, the most effective replication of ribosomes so far can copy RNA sequences up to 95 nucleotides long, which is not very long in terms of a gene, but is fairly long in terms of being able to self-replicate to be able to get this party started in terms of creating life from non-life. So the original world is very likely to have been an RNA world. So DNA-based life would have been preceded by RNA-based life, And in this world, RNA would serve as its own genetic molecule that not only carries information, but replicates itself. So it's able to create its own copies of itself. Now, as we know today, DNA replication is very complex and has a lot of editing that occurs at all times. And that means that the very first self-reproducing ribosome was probably not very efficient in terms of replication, meaning that it probably produced copies with lots of errors. It means that we have a ton of variants in a very short period of time, and the process of natural selection was likely to have acted on those errors to improve the function of the early ribosome. So it would have been an all-out arms race in these early stages of the RNA world. Eventually leading to increased speed and accuracy of replication, these ribosomes would reproduce, copy themselves, and displacing less efficient molecules. This is similar to what I was talking about previously when I said that no one in the class has an iPhone 4 anymore, right? And almost no one I know has an iPhone 2 or iPhone 3 because the 4 is more, it's just better, it's more efficient, it has better storage capability, etc. And so every time that the newest copy comes around, the older copy becomes obsolete. So over time, we get more and more efficient replication machinery, allowing the evolution of these molecules. And eventually, we end up with RNA becoming an intermediary between DNA and proteins instead of becoming its own RNA world, right? So we're not really sure exactly how that happened, but at some point, RNA slipped into the background, and DNA started taking over in terms of the information molecule, and RNA became an intermediary molecule between DNA and proteins. However, at this point where we're talking about RNA that is self-replicating, we are still not at the point of life, right? Just having a self-replicating molecule is not considered to be alive. In living cells, which is what we consider to be the smallest unit of life, we have to have a membrane that encloses the DNA or RNA inside, right? And so that means that we had to have some sort of vesicular agitation. What does that mean? Basically, what it means is that if water has protein in it and lipids in it, and it's being beaten on the shore, so we agitate it to simulate waves, the proteins and the lipids eventually form hollow vesicles. And this is going to be the precursor to the cell, right? And eventually we're going to end up with vesicles that have very defined outer boundaries that separate internal and external environments, and this is the definition of a cell, correct? And if we have the correct composition to the membranes, they actually can be very, very similar to that of what we consider to be cellular life today. And so this is demonstrating that early life would have been created by molecules that were in the water surrounded by these amphipathic molecules and proteins and lipids that then would have come together to make vesicles surrounding that DNA or RNA and allowed them to absorb material from the external environment, keep the internal environment separate from the external environment, grow, divide, replicate, etc. All of the things that are classically known as life on Earth today. Now, this would not have been, these are considered protocells. They're not exactly yet alive. This is the very yet first precursor to cells. They would have contained organic molecules, including the ribozymes that we just talked about, and again would have been the precursors to living cells. This is the very first step in which we have membranes sequestering molecules inside and keeping protection or protective layer from the external environment. Eventually, over time, the protocells would have developed the ability to divide and pass on copies of the ribosomes to their daughter protocells. The transition from protocell to living cell is a continuous process, one step at a time, with no defined boundary between one state and the next. But did it all really happen? And we've demonstrated by the experiments of Stanley Miller and others that it actually was capable of happening with our very basic building blocks in the primordial soup. So I'm going to go ahead and stop here, and we will pick this up in section 2.